Kingdom. When you look around, you will observe different animals with different structures and forms. As over a million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. Basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory, or reproductive systems. These features are used as the basis of animal classification, and some of them are discussed here. Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, that is, they exhibit cellular level of organization. Some division of labor activities occur among the cells. In coelentrates, the arrangement of cells is more complex. Here, the cells performing the same functions are arranged into tissues, hence, is called tissue level of organization. A still higher level of organization, that is, organ level, is exhibited by members of platyhelminths and other higher phyla, where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms, and chordates, organs have associated to form functional system, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ system is different group of animals. Organ system in different group of animals exhibit various patterns of complexities. For example, the digestive system in platyhelminths has only a single opening to the outside of the body that serves as both mouth and anus, and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types: open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and closed type, in which the blood is circulated through a series of vessels of varying diameters, arteries, veins, and capillaries. Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical, that is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of any when any plane passing through the central axis of the body divides the organism into two identical halves, it is called radial symmetry. Coelentrates, ctenophores, and echinoderms have this kind of body plane. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. Diploblastic and triploblastic organization. Animals in which the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. Example: coelentrates. An undifferentiated layer, mesoglea, is present in between the ectoderm and endoderm. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm, are called triploblastic animals. Platyhelminths to chordates. Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates. Example: annelids, mollusks, arthropods, echinoderms, hemichordates, and chordates. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called cirrocoelomates. Example: aschelminths. The animals in which the body cavity is absent are called acoelomates. Example: platyhelminths. Segmentation. In some animals, the body is externally and internally divided into segments with the serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates. Example: Porphyra to echinoderms.